You know, looking through this massive stack of microbiology and pharmacology research you sent over, it really hits you how much we tend to think of ourselves as these singular, independent beings.
Oh, absolutely. We just walk through the world feeling like one cohesive unit.
Right. You look in the mirror, you see one person. But the reality, and I guess the central theme of the notes we're diving into today, is that you are essentially a walking, breathing ecosystem.
Yeah, an invisible battlefield, real.
Exactly. And since you, the listener, are someone who clearly loves understanding the hidden mechanics of the world, we tailored this deep dive specifically for you. We're going to explore exactly what's happening in that microscopic war going on inside you right now.
It's a profound shift in perspective when you realize that we are quite literally just covered in microbes. And what really stands out in these sources is that our normal microbiota is, well, it's highly specific.
Oddly adapted too, right?
Right. It's not just a random assortment of bugs clinging to us. They have established territories.
The documentation shows it's like different neighborhoods have totally different demographics and they are fiercely territorial. Like on your skin and in your nose, Staphylococcus epidermidis absolutely dominates the landscape.
Yeah, they basically own that real estate. But then you travel down to the colon, and it's a completely different environment. Down there, Bacteroides fragilis vastly outnumbers E. coli.
Which is wild to think about. And in the vagina, lactobacillus acts as this incredible microbial guard, right?
Exactly. They take up the physical space and the local resources, and by doing that, they actively prevent the truly dangerous microbes from moving in and setting up shop.
Okay, let's unpack this. Because today's mission is all about what happens when that system fails. What happens when the wrong bugs breach the perimeter?
And maybe more importantly, how exactly do the pharmacological weapons we've invented actually hunt them down and destroy them?
Right, but to understand the weapons, we first have to map out the sheer bizarre variety of the enemies we're fighting, I think.
Definitely. Because microbes don't all operate by the same biological rules. Prions are a perfect starting point for this.
Oh man, the section in your notes on prions is wild.
Right. Structurally, they completely upend our standard definition of an infectious threat. Because, I mean, they aren't even alive.
Yeah, they aren't bacteria, they aren't viruses, they don't even have DNA. A prion is literally just a protein.
Just a single protein.
Eh.
Normally, we have these proteins in our bodies, especially in the central nervous system, called PRPC.
And normally they fold into an alpha helical structure, right? Which is basically like a flexible, coiled spring.
Exactly. But a prion disease happens when that protein misfolds into a beta-pleated form called PRPC. So instead of a coiled spring, it becomes this rigid, folded sheet of paper.
And that structural shift changes everything, doesn't it?
It is everything. Because our bodies rely on proteases, which are specific enzymes that act as cellular garbage disposals to break down old or damaged proteins.
But those proteases are only designed to cut the spring shape.
Exactly. When the protein folds into that rigid sheet shape, the proteases just can't latch onto it. It becomes completely resistant to degradation.
So it just sits there, unable to be cleared out. And the notes say this misfolded protein somehow bumps into normal proteins and forces them to misfold too.
Yeah, it's basically a cascading effect.
They're basically zombie proteins. One gets misfolded, it bites a normal one, turns that one into a rigid zombie, and they just keep piling up in the brain.
That is a terrifyingly accurate analogy.
Yeah.
And the mechanical result of that accumulation is what we call transmissible spongiform encephalopathies.
Which includes diseases like Creutzfeldt-Jakob disease or mad cow disease.
Right. Because these rigid sheets clump together and destroy neurons, the brain tissue literally develops physical holes. It resembles a sponge, which is what spongiform refers to.
Wow. And because they're just misfolded proteins, they survive extreme sterilizing procedures, right? Including standard hospital autoclaving.
Yeah, you just can't kill something that was never alive to begin with.
Thankfully, those prion diseases are incredibly rare. But if we look at the survival mechanics, prions survive by basically breaking the rules of biology.
Completely breaking them.
But the bugs that cause everyday food poisoning, on the other hand, survive because they don't even wait for you to eat them. They weaponize your food before it hits your plate.
Exactly. The battlefield dynamics are completely different there. Take Bacillus cereus, which is famous for contaminating reheated rice,
Yeah.
or Staphylococcus aureus in mayonnaise left out in the sun.
The bacteria are multiplying in the food and actively manufacturing preformed toxins, right?
Yes, they're basically leaving little chemical landmines.
Which explains that mnemonic in the research, food poisoning from reheated rice. Be serious. for be serious.
I love that one. It really sticks in your head.
But the key distinction here is that when you eat that rice, it's not the bacteria invading your stomach lining that makes you violently ill 20 minutes later.
Right. You're ingesting a toxic chemical payload they already built. It's a chemical hit and run. That's why it hits fast and ends fast.
Compare that hit-and-run tactic to organisms that cause bloody diarrhea, like Shigella or enterohemorrhagic E. coli O157HH7.
Yeah, those are much more invasive. They don't just leave a toxin in your food. They have to survive the stomach acid, reach the colonic mucosa, and physically invade the tissue.
Or, once inside, they produce Shiga toxin, according to the notes.
Right. And Shiga toxin doesn't just make you nauseous. It actually halts protein synthesis in your cells.
So it's physically dismantling your intestinal lining, which is why you get bloody dysentery.
Exactly. And it can enter the bloodstream and cause hemolytic uremic syndrome, or HUS.
Which sounds like a devastating complication.
It is. The toxin damages the lining of small blood vessels. As red blood cells try to pass through these damaged vessels, they are physically sheared apart.
And that's the hemolytic part of the syndrome?
Right. Those shredded cell fragments then travel to the kidneys and basically clog the microscopic filtration system. That causes acute renal failure, which is the uremic part.
It's terrifying how specific these attacks are. Microbes aren't just randomly flailing around. They have highly evolved strategies, and they target very specific anatomical niches.
And very specific demographics, too. It turns diagnostics into a high-stakes biological game of clue for doctors.
Because when a patient crashes, you don't have 3 days to wait for a lab culture to grow.
Exactly. You have to start empirical treatment immediately based entirely on statistical probability and epidemiological mapping. Meningitis is the classic example of this.
Because the organisms that cause inflammation of the brain's lining shift radically depending on the host's age.
Precisely. If a newborn baby develops meningitis, the statistical culprits are usually group B streptococcus or listeria.
And the notes point out that group B strep rates have actually plummeted because we actively screen pregnant women and use maternal antibiotics during labor.
Yeah, public health interventions really work there. But if a 40-year-old adult comes into the ER with a stiff neck and a fever, you aren't worried about group B strep at all.
You're hunting Streptococcus pneumonia and Neisseria meningitatis.
Exactly. And while the physician is prescribing antibiotics to cover those specific targets, they perform a lumbar puncture to analyze the cerebrospinal fluid, the CSF.
Right, because the biochemical debris in that fluid tells you instantly if you are fighting a bacterial army or a viral one.
Yes. If it's bacterial, the CSF will be flooded with PMNs, polymorphonuclear neutrophils, and the glucose levels in the fluid will be completely tanked.
But if it's viral, you'll see a surge in lymphocytes, and the glucose actually remains normal.
The mechanism behind that glucose drop is fascinating, really. Bacteria are living, dividing cells with their own independent metabolisms.
So they are literally consuming the free glucose in the spinal fluid for energy.
Right. Viruses, however, are just genetic material packaged in protein. They don't have their own metabolism.
They just hijack the host's cellular machinery to replicate so they don't need to consume the ambient sugar in the fluid.
Exactly. We see that same extreme niche targeting in pneumonia, too. The local environment completely dictates the invader.
Oh, like with cystic fibrosis patients. Their lungs produce an incredibly thick, sticky mucus, which creates a low-oxygen environment perfect for Pseudomonas to build biofilms.
Right. Or consider someone who overuses alcohol and loses their gag reflex. They might accidentally aspirate.
Pulling anaerobic bacteria straight from their own mouth, like Bacteroides or Fusobacterium, straight down into the lungs.
What's fascinating here is how this dictates the immediate counterattack. If a doctor suspects bacterial meningitis in an adult, they look at those probabilities and instantly deploy a combination of ceftriaxone and vancomycin.
So they are basically throwing a highly calibrated chemical net over the most likely targets before they even know the exact identity of the bug.
That's exactly what empirical treatment is.
Okay, so you've played the biological game of Clue, you've identified the likely suspect, and you've launched the counterattack. Now we get to the core of this whole deep dive.
The pharmacology part.
Yeah, how do we actually kill the bug without killing the human it's sitting inside of? And that brings us to targeting the bacterial cell wall.
It is the ideal structural vulnerability. I mean, human cells are encased in flexible cell membranes. Bacteria have membranes too, but they often live in harsh environments.
So they have to encase their membrane in a rigid, highly pressurized shell made of peptidoglycan.
Right. If you can compromise the structural integrity of that wall, The internal osmotic pressure of the bacteria is so immense that water rushes in and the bacterium physically explodes.
Wow, it's like popping a microscopic overinflated tire. And penicillin was the original weapon for this, wasn't it?
It was, but the molecular mechanism of how penicillin does this is just brilliant chemical mimicry.
It really is. The notes say that rigid peptidoglycan wall is made of long molecular chains, but to have any actual strength, those chains must be cross-linked together.
Yes, and the bacterial enzyme responsible for building those cross-links, the transpeptidase, looks for a very specific amino acid sequence to grab onto.
Dealanine, right? Or diala.
That's the one. And penicillin is a structural analog of diala. It essentially wears a disguise, so the enzyme thinks it's a structural building block.
So the transpeptidase grabs the penicillin, but penicillin irreversibly binds to the enzyme's active site, completely jamming it.
Exactly. The cross-linking stops, the wall loses its integrity, and the bacterium ruptures.
Well, wait, I want to push back on something in the notes here. Because penicillin doesn't work on everything anymore, we frequently use vancomycin, right?
Yes, it's the heavy hitter for drug-resistant bugs like MRSA.
But the sources state vancomycin also targets the cell wall, and it also targets the diala structure. If they're both targeting the exact same vulnerability, why use one over the other? Isn't that functionally redundant?
It sounds totally redundant on the surface, I know, but the biochemical mechanism is fundamentally different. Penicillin attacks the enzyme that is trying to build the wall.
Okay.
Vancomycin is a massive, bulky glycopeptide molecule. It doesn't bother with the enzyme at all. It binds directly to the diala precursors, the raw building materials themselves.
Ah, I see. So if penicillin is a saboteur that permanently breaks the bricklayer's trowel, vancomycin is like a giant tarp that physically covers up all the bricks so the bricklayer can't pick them up.
That is a highly accurate way to visualize it. Because vancomycin is so bulky, the enzyme physically cannot reach the diala site to create the crosslink.
That makes perfect sense.
However, because it's such a heavy-duty molecule, the deployment comes with risks, notably the vancomycin infusion reaction.
Right, the notes mention this causes a massive, diffuse red flushing all over the patient's body, which sounds like a severe allergic reaction, but it's actually not an allergy at all, is it?
No, not at all. It is a direct pharmacological effect. If you infuse vancomycin too rapidly into the bloodstream, the sheer concentration of the drug directly stimulates mast cells to dump massive amounts of histamine.
So it's literally a mechanical reaction to the speed of delivery.
Exactly. That's why it is highly preventable. You simply decrease the IV drip rate and administer antihistamines to block the receptors.
Here's where it gets really interesting, though. We know how to break the wall, but what if the bug doesn't rely on a standard cell wall? Or what if it's totally resistant?
But we can't pop the tire. We have to sneak inside the cell itself and jam their microscopic manufacturing plant.
Right. We are talking about protein and DNA synthesis inhibitors, and the central challenge in pharmacology is selective toxicity, right?
Yes, exactly. How do you poison the bacterial factory without poisoning the human factory right next to it?
It's like sending a saboteur into an industrial park who is only trained to dismantle one specific model of a machine. If they see a human machine, they don't know how to break it, but if they see a bacterial machine, they completely wreck it.
The machines in this scenario are ribosomes, the complex molecular engines that translate genetic code into actual proteins. Human ribosomes are categorized by their sedimentation rate as 80S.
And bacterial ribosomes are slightly smaller, right? Categorized as 70S.
Yes, and they are built from two subunits, a 30S and a 50S piece. Because the physical shape of a 70S ribosome is distinct from an 80S ribosome, we can design drugs that only dock onto the bacterial versions.
The sources include that classic medical school mnemonic for this, by AT30, CCEL at 50.
It's a lifesaver for students.
So the A and T are aminoglycosides and tetracyclines, which specifically jam the 30S subunit, and the CCEL, chloramphenicol, clindamycin, erythromycin, and linezolid, they bind to the 50S subunit.
Right. Erythromycin is a macrolide, which is what a standard Z-Pak is. It literally physically blocks the exit tunnel of the 50S subunit, so the newly built protein chain can't leave the factory.
That's incredible. But as precise as these saboteurs are, there's always the risk of collateral damage. Tetracyclines are a prime example in the notes.
Oh, absolutely. They are incredibly effective, but they have a strong chemical affinity for chelating or binding to divalent cations like calcium.
Which explains the two major rules with tetracyclines. First, if you give them to young children, the drug binds to the calcium in their developing teeth, causing permanent discoloration.
Classic side effect.
And second, you can't take them with a glass of milk. because if you ingest dairy, the drug binds to the calcium in your gut, forming an insoluble complex that your body can't absorb.
You're effectively neutralizing the antibiotic before it ever reaches the bloodstream.
It really highlights the chemical reality that these drugs don't exist in a vacuum. They interact with the entire biochemical environment of the host.
And sometimes we don't even fully understand those interactions yet. Look at fluoroquinolones like ciprofloxacin.
Yeah, that's a fascinating one.
These don't target the ribosome at all. They target DNA gyrase,
Hmm.
which is the enzyme bacteria use to unwind their DNA for replication. You jam the unwinder, the DNA gets tangled, the bacteria dies.
Sounds perfect. Right.
But bizarrely, a known side effect in humans is ruptured Achilles tendon. Why on earth would a DNA unwinder drug cause your heel to snap? It is a remarkable and severe adverse effect, particularly in older patients or those on corticosteroids. The exact mechanism is still being mapped out, actually.
But there's a working theory in the sources, right?
Yes, the current understanding is that fluoroquinolones upregulate matrix metalloproteinases in human tissue. These are enzymes that chew up collagen.
Wow. So while the drug is busy fragmenting bacterial DNA, it is simultaneously degrading the collagen fibrils in the patient's tendons,
Exactly, just weakening them until they literally snap under tension.
which is an incredible transition into the escalating arms race of this whole ecosystem. Because bacteria aren't just sitting ducks letting us pop their walls and jam their DNA, they are actively evolving countermeasures.
Right, the classic evolutionary response is their defense against penicillin. Bacteria developed an enzyme called beta-lactamase.
Because penicillin's molecular payload relies on a core structure called a beta-lactam ring, right?
Precisely. The bacteria evolved an enzyme that acts as a pair of highly specific molecular scissors, designed to cleave that ring open, rendering the drug completely inert.
So they invent a chemical shield, and we invent a countermeasure, beta-lactamase inhibitors. Things like clavulanic acid or tazobactam.
The acronym in the notes is CAST, yeah.
Right, and we literally package these inhibitors in the exact same pill as the antibiotic. The bacteria send out their scissors to destroy the antibiotic, but our inhibitor acts as a sacrificial decoy.
It permanently binds the scissors, leaving the actual antibiotic free to slip past and destroy the cell wall.
It is literal microscopic warfare. But as complex as bacterial warfare is, the sources dedicate a significant portion to viral warfare, which operates on an entirely different paradigm.
Because you can't just target a viral factory. They don't have one. They hijack our factories.
Which means any attempt to stop viral replication inherently risks shutting down the host's own cellular machinery.
And that was the monumental hurdle in developing treatments for HIV and hepatitis C.
But looking at how we solved it with HIV is staggering. We use antiretroviral therapy, or RT. A core component of this is NRTI's nucleoside reverse transcriptase inhibitors like tenofovir or zidovudine.
And they work by essentially feeding the virus faulty building blocks. Let's look closely at that mechanism because it's brilliant.
Okay.
When HIV enters a human T cell, it uses an enzyme called reverse transcriptase to build viral DNA. It does this by stringing together nucleotides, much like snapping Lego blocks together.
And NRTIs are structurally identical to normal nucleotides, but they are missing one vital component, a 3' hydroxyl group.
Exactly. So it's exactly like a Lego piece that has the bottom holes to snap onto the piece below it, but it's completely smooth on top. It's missing the studs.
Oh, wow. So when the viral enzyme grabs the NRTI and attaches it to the growing DNA chain, there's chemically nowhere to attach the next piece.
The chain is irreversibly terminated. Viral replication just halts instantly.
And we don't just rely on that one trick either. We hit them from every angle. We use protease inhibitors to stop the viral proteins from being cut into their mature functional shapes.
Right.
We use entry inhibitors like Maraviroc, which physically block the virus from docking onto the CCR5 receptors on the outside of our T cells in the first place.
If we connect this to the bigger picture, the development of these multi-drug therapies represents a monumental shift in pharmacology. We moved from merely trying to suppress the viral load to structurally dismantling its entire life cycle at multiple simultaneous biochemical choke points.
And with hepatitis C, the sources highlight that we went from just managing the disease to literally curing it. We developed drugs that target the highly specific non-structural proteins the hepatitis C virus uses to replicate.
Yes, the NS5A, NS5B, and NS34A inhibitors. The mechanism of action for drugs like sofosbuvir, which is an NS5B inhibitor, is a triumph of targeted design.
Because it specifically targets the viral RNA polymerase, the enzyme the virus uses to copy its genetic blueprint, right? Yep.
Exactly. So Fosabuver acts as a defective substrate for that specific enzyme, causing chain termination. It doesn't just slow the virus down, it completely paralyzes the replication machinery inside the human liver until the existing virus is entirely cleared from the body. It is a true, definitive cure.
It is unbelievable when you zoom out and look at all of this. For you, the listener, who brought us this incredible stack of research, we've journeyed from the unkillable, rigid sheets of prion proteins through the high-stakes biological clue game of mapping meningitis targets.
All the way to popping bacterial tires and feeding viruses smooth Lego blocks.
Yeah. Systems microbiology isn't just about memorizing bugs. It's about understanding the exact intersection of where a microbe thrives, the mechanical machinery it uses to survive, and the highly specific chemical wrench we've designed to throw into those gears.
It really changes how you see medicine.
Absolutely. The next time you get a prescription for a standard Z-Pack or some amoxicillin, you won't just see a pill. You'll know you are holding a highly targeted molecular saboteur, engineered over decades to hunt down a 70s ribosome or permanently jam a cell wall builder.
But we have to recognize that this microscopic ecosystem is incredibly dynamic, and the war is far from over.
Right, they are fighting back.
Exactly. The microbes are already adapting to our most advanced structural analogs. Consider vancomycin, our heavy hitter that binds to the Diella building blocks. Certain Enterococci bacteria have already mutated their genetic code to change that target sequence from Diella to Diella-Lac.
Just replacing one amino acid with a lactate molecule.
Yes, and that single tiny molecular shift means vancomycin's bulky tarp has nothing to grip. It becomes completely useless.
Wow.
Furthermore, bacteria are actively sharing genetic blueprints for plasmid-encoded efflux pumps. These are literal microscopic sump pumps embedded in their membranes that physically spit our drugs back out of the cell as fast as they enter.
That is insane.
It is, which leaves us with a critical ongoing question to consider. If these organisms can spontaneously rewrite their own genetic machinery to outsmart our most ingenious pharmacological weapons, what happens when we simply run out of unique cellular vulnerabilities to exploit?
Are we nearing the inevitable end of the antibiotic era, or are we just standing on the cusp of an entirely new, even more complex phase of this invisible battlefield? Something to think about until our next deep dive.